I'm Dr. Shinomol George from the Department of Life Sciences, and I'll be talking about auxins, its biosynthesis and functions under the course Plant Physiology. There are 5 major types of plant hormones: auxins, cytokinins, gibberellins, ethylene and abscisic acid. Auxin is part of cell growth and expansion and is usually found in the parts of the plant that are actively growing with the highest concentration in the primary stems. Auxins are most effective when they are partnered with the another hormone, that is cytokinins, working alongside to stimulate fast and early nodal root development. This is one of the first major plant hormones that were discovered, and they come under the indole-3 acetic acid, which is the most common naturally occurring plant hormone of the auxin class. And common functions of auxins include root formations responsible for apical dominance and phototropism, promotes xylem differentiation, retards leaf abscission and promotes fruit growth. And coming to types of auxins, the naturally occurring auxins are indole-3 acetic acid, 4-chloro-indole-3 acetic acid, phenyl acetic acid and indole-3 butyric acid. And synthetic auxins are like 2,4-dichlorophenoxyacetic acid, alpha-naphthalene acetic acid, 2,4,5-trichlorophenoxyacetic acid, 2-methoxy-3,6-dichlorobenzoic acid and 4-amino-3,5,6-trichloropicolinic acid. Then comes the free and bound auxins. The free auxins are readily extractable and they are immediately utilizable in growth, whereas the bound auxins, which are liberated from tissues when they are subjected to enzymolysis, hydrolysis or autolysis, forms the storage form of this hormone. They are synthesized in the seed maturation stage and hydrolyzed during the seed germination stage. And coming to auxin biosynthesis, there are 2 major pathways of indole acetic acid biosynthesis that have been proposed in plants. One is the tryptophan dependent pathway and the second is the tryptophan independent pathway. So auxin biosynthesis mechanisms can be majorly 4, that is indole-3 pyruvic acid pathway, tryptamine pathway, indole-3 acetonitrile pathway and indole-3 acetamide pathway. Coming to the IPA pathway or indole-3 pyruvic acid pathway, the tryptophan loses the amino group by deamination and indole acetic acid, which then loses carbon dioxide to form indole-3 acetaldehyde. This oxidation of this indole-3 acetaldehyde results in the formation of indole-3 acetic acid. Then coming to a TAM pathway, it occurs in those species which lack the IPA pathway. The tryptophan is first of all decarboxylated from tryptamine, which is then oxidized as well as deaminated to form indole acetaldehyde. And this non-Hebal et al. In 1993 they found that both IPA as well as TAM pathway are present in tomato, which is a rare thing. The IAN pathway, the indole-3 acetamide pathway and the IAN and other compounds have been found primarily in the Brassicaceae family, and this pathway has not been considered to be of general importance. Tryptophan is here converted to indole-3 acetaldoxime by an enzyme activity that has been detected in several plant species, and indole-3 acetaldoxime conversion to indole-3 acetonitrile has been demonstrated in plasma membranes of Chinese cabbage, and nitrulase is an enzyme that converts this indole-3 acetonitrile to indole-3 acetic acid. So this diagram shows a combined IPA, TAM and IAN pathway. You can see IPA on the IPA pathway, tryptophan on deamination gives indole-3 pyruvic acid. Decarboxylation will lead to indole-3 acetaldehyde, which is oxidized to indole-3 acetic acid. In the TAM pathway, which are all these 3 pathways are the tryptophan dependent pathway. So in the TAM pathway, decarboxylation leads to the formation of tryptamine, followed by oxidation and deamination leading to indole-3 acetaldehyde formation, which is further oxidized to indole-3 acetic acid. In the IAN pathway, indole-3 acetaldoxime formed is converted to indole-3 acetonitrile and finally to indole-3 acetic acid. The auxin biosynthesis as such is an extremely complex process which starts in the chloroplast and ends in the cytoplasm. So it is likely that multiple pathways contribute to de novo auxin production, and indole 3 acetic acid can also be released from indole 3 acetic acid conjugates by hydrolytic cleavage of indole 3 acetic acid amino acid conjugates, or IAA sugar conjugates, or IAA methyl ester conjugates. Here in this diagram you can see in the chloroplast, from the anthranilate tryptophan formation takes place, and this tryptophan enters into the cytoplasm and the tryptophan dependent pathways will happen. Whereas for the tryptophan independent pathway, the anthranilate is getting converted to indole 3 glycerol phosphate, which is entering into cytoplasm and follows the tryptophan independent pathway and gets converted to indole 3 acetic acid. Thank you.